Hello, class. So today we're going to be picking up from where we left off last time. And we're going to be thinking about how we get from genes that have been under selection to those traits in question. And the reason why we need to do this is because traits end up being a lot more complicated than the genes that we've been thinking about. So far, we've established that the gene's eye view is the helpful level to think about how natural selection acts on sexually reproducing species, because individuals can be are going to have different genes in them between parents and their offspring, and also different genes within an individual are going to have different fitness interests. But now we're going to be scrutinizing the complicated way that genes can have effects on the traits of organisms. And we looked at an example of this already. If you remember back to the BRCA mutations that we talked about as risk factors for breast cancer, and how counterintuitively the gene that mutated is not about breasts per se, not about the construction of breast tissue, but rather about tumor suppression. This mistake is possible because there's an infinite number of ways to carve up the nature of any species, human nature, into traits. How do we know which carving to use when we're understanding what genes do? If you remember that allegory of the blind men and the elephant, that's an example of what can happen when you get the carving wrong, right? You're looking at something that has pieces that make sense when you look at it from a particular angle, but looking at it from any other angle, and the pieces that you draw no longer make sense given what it actually is. The question is, how big is this problem? How much does the carving that you use matter? Here's an example I like to use where understanding what the trait is can have dramatic effects. These little creatures are known as sea squirts or ascidians, and here's a group of them perched on this rock. A funny thing about sea squirts is that some of them have brains. That's the long, skinny, yellow stripe down the middle. That's their nerve cord. And some of them don't. That one doesn't have a brain. If you wanted to try to understand how this happened, you might go looking for brain genes, right? Genes that the brain havers have that the brain not havers don't have. That seems like it would be an intuitive way to go about it, right? If you have a brain, you should have genes for a brain. And if you don't have a brain, you may not have genes for that brain. The problem is that all of these creatures have the same genes. It turns out that young sea squirts have brains, while by the time they get to be adults, they don't. Sea squirts, what they are, how they make a living, is as a filter feeder. They eat nutrients and things out of the water that they're filtering. When they're young, they need to search around for a good place to latch onto that will provide a steady stream of food for the rest of their lives. And so when they're young, they have to behave, they have to sense, and they have to move. But once they attach to the rock, they don't need to sense anymore. They don't need to find, and they certainly don't need to move because they're stuck. And what these creatures do is they actually digest their brain and they reconfigure that tissue into sex organs, organs that produce gametes, sperm and eggs. That's a pretty unique and complicated trait, right? It's not just a brain, it's that whole system of creating a brain, digesting it, and turning it into something else, right? If you wanted to find the genes involved, it's going to be helpful to actually understand what the trait actually is, as opposed to simply looking for genes that the creature on the right has, that the creature on the left doesn't have. Unfortunately, this problem, or this general approach of looking at who has the trait and what genes differ between them and others, is really common. For example, people are often aware that there's variance in bodies and behavior. For example, some of you are tall, some of you are short. That's variation in your bodies or your phenotype. Some of you are hyperactive and some of you are very calm. That's variation in your behavior. And ever since Mendel, we've been mapping out variance in genes. Putting those data sets together, we can find correlations between the genes and the traits. So we can model how much of that variability in the trait is accounted for by variance in the genes. And this is the concept of heritability. And for decades, the field of behavior genetics has been focused on studying. Most people have a fairly intuitive understanding of heritability and what it means, but to examine critically what it means and what it doesn't mean, let's look at the formal mathematical definition. Heritability starts with a model that says that all of the variance in a trait is the total of the variance that's due to genes, the variance that's caused by environmental differences, and the variance that's due to different genes producing different effects in different environments. That is a gene by environment interaction. And so each of those V terms is simply the phrase variance due to. So putting that into concrete terms, let's say you have a population where some people have blonde, brown, black, or red hair. That's variance in the trait. And if you look at all the variability in the hair color across a population, that would be the total amount of variability to be explained. This model says that we can understand variance coming from different people having different genes, like hair color running in different families. So blonde parents being more likely to have blonde kids, that would be variance due to genes. Or the variance could come from environmental sources, like some people dye their hair black or dye their hair blonde. And then finally, there could be an environmental by gene interaction where some genes might lead to different hair color in different environments. Once you model the variability this way, a simple mathematical re-expression lets you take the term for variance due to genes, that V genes, and divide it by all of the variance in the trait. That makes it a proportion. It gives you a proportion of variance in the trait that's due to variance in the genes. And as a proportion, this can range from 0, meaning none of the variance in the trait is due to variance in the genes, to 1, meaning that all of the variance in the trait is due to variance in the genes. So far, so good. That seems to tell us something that we want to know, right? Okay, well, how do we estimate these things? Well, to get to variance due to genes, there are a few techniques. You could compare different kinds of siblings, like identical twins, fraternal twins, etc., and measure how much more similar closer related siblings are to each other, given that they share more genes. If you haven't learned this much already, there's actually quite a few options here. Starting off with the most related sets of siblings, you have what are called monozygotic or identical twins. Monozygotic means mono means one, they came from the same zygote, that same initial fusing of sperm and egg. So they came from a sperm and an egg, and then it split into 2 embryos. But within identical twins, within monozygotic twins, you can have variance in how much of the uterine or environment during pregnancy that they shared. They could share the same placenta. That's the organ that attaches the umbilical cord to the uterus. That is, they could be monochorionic. Chorion is just a fancy term for placenta. And they can be mono or diamniotic. The amnion is the sac that surrounds the developing fetus. And so you could have identical twins that are sharing the same placenta or have different placentas. You can have identical twins that share the same sac or that have different sacs. And those various combinations are going to mean that across these sets of twins, they all have the same genetic similarity, but because of differences in how those placentas can act and differences in the environment within each of those sacs, they can experience some variance in the environment that they experience. Next, we have dizygotic or fraternal, di meaning 2. They came from 2 zygotes. So this is 2 sperm, 2 eggs that fused at the same time. The pregnancy happened at the same time. The pregnancies happened at the same time, but they're basically just like full siblings. They're full siblings that happened to share the same uterus at the same time. And so if you think about the difference between dizygotic twins and full siblings, they have the same amount of genetic similarity, but the dizygotic twins have more shared environment in common than the full sibs, because the dizygotic twins shared all of those experiences during that pregnancy because they were in that pregnancy together. Then comparing full sibs to half sibs, sib is just short for sibling. A half sibling is a brother or sister that only shares one parent with you, while a full sibling is a brother or sister who shares both parents with you. So compared to a full sibling, a half sibling has a smaller proportion of genes in common. And then finally, we could think about step siblings, siblings that you're not related to genetically. Your parents married after each of you were born, and so you have different parents genetically, but you happen to be raised in the same house for at least a portion of time, and so you share some environment. You share some of the same environmental experiences that maybe kids who were raised in other families don't. Now, often, that's the thing that people are most interested in, that variance due to genes. And that's the thing that they try to get the most precision in their estimate for. And if you think about the overall component, variance in the trait, that's the thing that's the easiest to see, because you don't have to look at what caused what. You can just look to see who has black hair, who has blonde hair, what's the variability across the population. And so given that you have variance in the trait and variance in genes, you can just treat these other terms as basically being error. That's the rest of the variance that you're not trying to explain most of the time. So let's look at a simple twin study to see how this can be done to actually estimate variability. Sorry, so estimate heritability. Monozygotic, or MZ twins, share, for all intents and purposes, 100% of their genes. They have 100% genes in common because they came from the same zygote. Whereas dizygotic twins are basically just like full siblings. They only share as many genes in common as full siblings do, so about 50% genes in common. Say that you ran a study where you compared the similarity of monozygotic twins to each other and dizygotic twins to each other for some trait, like, say, intelligence. And you found that when one twin was very smart, their monozygotic twin was also very smart, that there was a high relationship between the intelligence of one monozygotic twin with his co-twin, but that having that same intelligence didn't predict quite as much for the dizygotic twin. They had more instances of smart twin, less smart twin, very unsmart twin, more smart twin, less of a relationship for the dizygotic twins, closer relationship, more correlation for the monozygotic twins. Concordance is simply the proportion where you have twins that match on the trait divided by the total number of twins that you're considering. So that 0.8 for the monozygotic twins would be 80% of them shared the trait. And for the dizygotic twins, that would be 60% of them shared the trait. And only 40% of them had differences. What we can do from that is we can say, well, when you go from being a dizygotic twin to a monozygotic twin, you're going from having 50% of your genes in common to having 100% of your genes in common. That's like a gain of 50% genetic similarity. And what that did, going from 50% to 100%, was that it gave you a gain in concordance of about 20%, 0.2. Therefore, if we wanted to think about how much more similar monozygotic twins would be compared to simply unrelated people, that is the entire effect of variance due to genes, all we would have to do is double that, right? Because going from unrelated, 0% genes in common, to monozygotic twins, 100% genes in common, that would simply be an estimate of that 20% increase in concordance you get going from DZ to MZ, and then you would just guess that that was consistent with what you get from going from 0% to 50%. And so that 40% gain in concordance is actually one of the simplest ways of getting to heritability. What that would say is that for that trait, let's say it was intelligence, that the heritability of intelligence was 40%, that 40% of the differences in intelligence could be explained by differences in genes, if that was the results of the study. So that's heritability in a nutshell. And using methods like these, researchers have found lots of traits that have substantial levels of heritability. Height is around 80% heritable. Weight is about 70% heritable. Intelligence is actually about 50% heritable. Lots of personality traits are in that 50% heritable range. I want you to reflect on that for a minute. Think about what you think that means. Most people have intuitions. They feel like that means something. And that's why news reports, study finds that intelligence is 50% due to your genes. Those studies, those news reports are popular. We feel like heritability means something that we care about and that we want to know. Imagine that you learned that a trait like, say, hand-eye coordination had a heritability of 75%. What does that tell you about it? Would you want to say that hand-eye coordination is 75% genetic? Would you want to say that hand-eye coordination is mostly hardwired by our genes and hard to change? These are some of the most common things that people think heritability means. People report that, well, if intelligence is due to your genes, then what's the point of sending kids to school? Because it's just about their genes, not about their schooling. We're going to tackle these intuitions because when most people hear about a heritability statistic, that is what they think it implies. And the big problem with that idea of heritability is that it is just systematically misunderstood, not only by the public, but also by the experts. And it leads to a lot of false inferences, false ideas. Like this one. Most people think that heritability reflects the genetic basis of a trait, such that if heritability is high, the trait is mostly genetic. And if the heritability is low, that means the trait is mostly environmental. And without knowing more, that sounds reasonable, right? But consider the arrowleaf plant. The arrowleaf is an interesting plant because it grows differently depending on how much water it gets. If it grows in a really kind of moist environment, like near a river or a stream, you get these short, stubby stems with broad leaves. In contrast, if it grows in a very dry location, you get these tall stems with short, thin leaves. That's variation in the trait, right? So think about it. What would the heritability of leaf shape be for the arrowleaf plant? Would anyone like to take a guess? If you had to guess if it was 0 or 1, what would you say? .5, splitting the difference. Well, think about it. All of these plants have the same genes. It's not about whether the seeds had one gene or another. It's about whether the seed was planted in a wet environment or a dry environment. So all of that variance in the trait is caused by how much water is in the environment, not by what genes the plant has. And so the heritability would be 0. Okay, so we can mathematically get to the idea that heritability for this trait would be 0. But does that 0 heritability mean that leaf shape doesn't have a genetic basis? Does it mean that there aren't genes for that leaf shape? Of course not, right? The plant wouldn't grow leaves at all if it didn't have genes that did that. And different plants that have different genes don't do this calibration to the local water level, right? They don't grow different kinds of leaves in different levels of moisture. And you don't grow different kinds of leaves in different kinds of moisture because you don't have those genes, right? Of course, that trait has a genetic basis, right? Heritability clearly doesn't pick out the genetic basis of a trait. And actually, the really non-intuitive thing to most people is that adaptations are going to show little heritability. Why? Well, if you think about how natural selection acts on different variations in a gene, when some of those perform better and the others perform worse, the worse performing ones are going to disappear and the better performing ones are going to spread, basically eliminating the variance in the trait. To the extent that those differences are actually differences in how well they work, we should expect natural selection to eliminate all of the differences that are due to genes. Because if there's differences that are due to genes, that's natural selection is going to be acting on those differences. That's the whole process of fixation. And so while most people think that if natural selection created that trait in us, we should find that it has high heritability, it's actually, in most cases, going to be the opposite. If natural selection has acted on a trait, we should expect there to be very low heritability. How about this one? People usually think that when you study the heritability of a trait in one population, that tells you something about the trait in general, that the heritability that you find is independent of the population that you study. And that's generally true in science. You study a sample to learn about the larger population, right? You study one sample of people to learn about something in people generally. The problem is that heritability is not the kind of statistic that does that. It's a descriptive statistic, not an inferential statistic. If you've taken inferential statistics in your coursework, this will make sense. And if you haven't, don't worry about it. But when we build that formula to understand heritability, we didn't build it in such a way that gives us the likelihood of observing a given level of heritability. If we sample from a population that has a specific level of heritability under that hypothesis about what the population was like, right? We didn't create a sampling distribution of heritabilities to understand the probability of getting a sample like we got. We're simply describing the sample that we have. Let's get more concrete, and this one everyone should get. Imagine that we have a set of identical twins where one of those twins, one member of each twin set, was raised in Boston, and the other member was raised with a group of hunter-gatherers in Botswana. It turns out that when you study people's vision, as we saw in the previous lecture, that you see that in industrial countries like the US, where people tend to spend a lot of time focusing on things near their face, like cell phones, a lot of people need to end up wearing glasses. And that who has glasses, who needs glasses, also tends to run in families. You can probably see this in, families that that parents, if the parents have glasses, it's much more likely that the kids needed glasses too. That is, if we think about, say, 5 different versions of a gene, 5 different alleles, G1, G2, G3, G4, G5, we would probably see in a country like Boston, in a place like Boston, in an industrialized country where people spend a lot of time reading and looking at things close by, that there would be variance in reading ability, variance in their vision, the proportion of people who have 20/20 vision, that's due to which genetic variance that you have. But if you think about that population of hunter-gatherers, they're not spending time doing that close focusing. They don't have cell phones they're looking at. They don't have books they're reading. They're spending their time looking farther away. And so all of these genetic variants, regardless of which one makes you more susceptible to developing poor eyesight, they're all doing pretty well. And so if we looked in Boston, we would find that what we observed as the differences between versions of genes and how well they performed doesn't extend to a different population. In fact, in this population, we would make an estimate that there was no heritability at all, because all of those differences in genes didn't contribute to any differences in vision. So this is exactly why we can't think about heritability being population independent. What we observe within a population, say we do a heritability study in a place where it's very easy to collect data. Like if I'm a researcher at Harvard collecting data in the local Boston area. What I've learned in my heritability study here, it actually doesn't tell me what heritability will be like around the world. Even though when you see these news reports that say intelligence is 50% genetic or eyesight is 90% genetic, they're not saying eyesight in Boston, right? Or intelligence in New York, or handedness in Shanghai, right? They're saying handedness in general, intelligence in general. And the problem is, that's not true. Okay, so heritability doesn't identify a trait's genetic basis, and it doesn't tell us something outside the population of study, but surely it must tell us that the trait is being genetically controlled in this population. If you can predict 100% of the variance in a trait by knowing what version of a gene that people have, that seems to imply that the trait is very rigid, hard to change, maybe even impossible to change. Is that correct? Well, think back a few lectures to our example with phenylalanine hydroxylase. This was that gene that turned phenylalanine into tyrosine, unless you had a defective phenylalanine hydroxylase gene, meaning that you no longer make that enzyme, and so you have that buildup of phenylalanine, and you develop that disorder, phenylketonuria, PKU. If you inherit a double mutation in this gene, such that you don't have that enzyme, you end up with this dangerous disorder, PKU. Given this information, what do you think the heritability of PKU be if we calculated it? Should it be 1? Should it be 0.5? Should it be 0? It's coming up on the slide, so you already have the answer, but very high, right? We're looking at the differences of who has PKU and who doesn't, and it's essentially only who has this double dose of these genes. But PKU is anything but hard to treat. In fact, it's very easily treated. We understand how it works. We understand what the problem is. And so we can either manufacture that enzyme and give it to people as like a pill so that their body has the enzyme, or you can restrict or limit the amount of phenylalanine that you eat in your diet. For example, just like on that can of Diet Coke, where you see the warning that says, attention, phenylketonurics contains phenylalanine, if you had PKU, you could avoid drinking things like Diet Coke, and you wouldn't have that problem. So high heritability doesn't even mean that a trait is genetically determined, hard to change. Okay, so we've ruled out the inference that heritability tells us whether a trait is genetic, whether it tells us something about the trait in other populations, or tells us if a trait is genetically determined. But surely if heritability means anything, it must be revealing the size of what you could call the genetic component of the trait. And that seems to simply follow from the way we calculate it, right? We take that proportion of the variance that's due to variance in genes, and if there's a proportion that's due to genes out of all of the variance, that's the amount that's genetic, right? Like intelligence, for example. Most estimates of heritability in intelligence put it around 50%. Does that mean that intelligence is 50% due to genes and 50% due to the environment? Think about the intelligence of the smartest person you know. Say this is a bar graph where height is intelligence. And the taller the bar graph is, the smarter that person is. And so think about that bar representing the absolute smartest person you know. Now think about the least intelligent person you know. Sorry, those should have happened in the other order. The taller one is the most intelligent. The lower one is the least intelligent. The reason why that isn't down at 0 is because there's other ways to have different genes, right? There are frogs, and frogs are much less intelligent than the least intelligent person. There's bacteria. There's the absence of those genes, like a rock. There's a lot less intelligence out there than the least intelligent person, particularly the least intelligent person in your study. When we're looking at heritability, all we're considering is the variation in the trait. That is, we're only looking at the portion of the trait that varies between people. We're not looking at the proportion of the trait that is common across people. And so what heritability is doing is it's saying within that range of variance, how much of that variance is associated with variance due to genes. It's only a proportion of the variance that exists between people, not to any extent that the trait is common across people. And if we think about natural selection and what it should be doing, natural selection is going to be strongly shaping complex adaptations so that they're invariant across people, right? Because anytime that there's a variant that works less well, natural selection is going to be acting on removing it from the population. That's going to be creating big pools of similarity across a population. Heritability is simply not a tool that tells you anything about how much the trait is genetic. Simply how much of the variance that you observe is due to variable genetics. And if that variance is actually like important variance that natural selection might care about, that's likely to be very small, a small amount of the total variance in the trait. It's only when that variance is kind of neutral with respect to natural selection doesn't really act on one versus the other because they perform relatively the same. That's the case where you could get substantial variability in the trait. But what that means is that heritability is mostly studying those things that natural selection hasn't selected the differences between. And that's really counterintuitive, right? Because that says that the things that you're studying as adaptations is not what behavior geneticists are studying, that you're studying two different classes of thing. So heritability does not tell you the genetic component of a trait. What is it then? At this point, you're probably confused as to what heritability actually is. The one thing it tells us is the variance in a sample in their trait level that's related to variance in that sample's genetics. This is strictly a property of a sample. And I emphasize that to contrast it with the final false inference that people often make. Heritability is not about a trait in an individual. You can't take a person and say that 50% of their intelligence is due to their genes and 50% due to their environments. That isn't what heritability says at all. It would be like trying to say it for a rectangle that some proportion was due to its height and some proportion due to its width. If you didn't have width, you wouldn't have area. And if you didn't have height, you wouldn't have area. And so the area is necessarily jointly determined by height and width. It's the exact same thing with any individual. All individuals are the joint product of their genes and their environment in a way that you can't separate. I think part of the persistent misunderstanding of what heritability means is that we often conflate the meaning. We think that it means something like simple heredity or simply inheritability, the ability of some trait to be passed on through generations. And this is like what we want to know, right? We want to know what's the probability, say, that an offspring will have a gene, the vertical line means given that a parent has that gene. So what's the likelihood of that gene being passed on? Or what's the probability that an offspring ends up with a trait, like having black hair, given that the parent has that trait, that the parent has black hair? But heritability doesn't actually tell us that. Knowing this requires knowing the actual mechanism. How do those genes work in those environments? You need to actually know how that works on a computational level. Simply looking at the heritability doesn't give you that. So how do you prevent yourself from making these false inferences going forward, particularly when you leave this class? The challenge is to look beyond the variance in the trait. You have to consider the entire adaptation in which that variance is expressed. And when you do that, it's much easier to avoid those mistakes. The challenge is to resist what Simon Baron-Cohen called instinct blindness. That's our tendency to find things that we do easily, to think that those things reflect something simple in our brains or in our genes, which causes us to ignore how complex they can be under the surface. This was our analogy for vision. But actually, Simon Baron-Cohen wasn't the first one to point this out. William James, the father of psychology in the United States, actually made this case all the way back in the 1890s in his book, Principles of Psychology. He wrote, "It takes a mind debauched." Debauched means kind of like ruined or made impure. It takes a mind debauched by learning to carry the process of making the natural seem strange so far as to ask for the why of any instinctive human act. To the metaphysician alone, a metaphysician was the old word for scientist, to the metaphysician alone can such questions occur as, why do we smile when pleased and not scowl? Scowl is another word for frown. Why are we unable to talk to a crowd as we talk to a single friend? Why does a particular maiden turn our wits so upside down? The common man can only say, of course we smile. Of course our heart palpitates at the sight of the crowd. Of course we love the maiden, that beautiful soul clad in that perfect form, so palpably and fragrantly made for all eternity to be loved. And so probably does each animal feel about the particular things it tends to do in the presence of particular objects, right? To the lion, it's the lioness, which is made to be loved. To the bear, the she-bear. To the broody hen, broody means just had eggs, the notion would probably seem monstrous that there should be a creature in the world to whom a nest full of eggs was not the utterly fascinating and precious and never to be too much sat upon object, which it is to her. Thus, we may be sure that however mysterious some animals' instincts may appear to us, our instincts will appear no less mysterious to them. This is the mindset that we have to get ourselves into in order to be able to see past the simple variance. So how do we think about that underlying similarity between people? That is, how do we get to an understanding beyond using something like behavior genetics to think about the genetics of what we all have in common? And why would we even expect there to be a commonness among members of a species in the first place? Why should there be a typical species nature, say for humans, such that you could say that there is a human nature? Last week, we explored the selection pressure of how pathogens led our far distant ancestors down the road to sexual reproduction. And that fact actually gave us the perspective that we needed to realize that genes in the same individual, even on the same chromosome, can have different fitness interests. And this intragenomic conflict clarified why we need to take a gene's eye view when thinking about selection pressures. Now we're going to think about another major implication of sexual reproduction. And this one is going to lead to the conclusion that adaptations must be universal within a species, and that we should expect a species typical nature. To get there, start with the fact that complex traits are made out of coordinated effects of a whole suite of genes, right? For something like a hand, you don't get that just on the basis of one gene acting alone. It takes a whole a lot of genes all working together in order to make that happen. So far, so good. But now think about what happens during sexual reproduction. Sexual reproduction takes those big sets of genes in the parent that they came from and worked together so well, and it rips them apart and mixes them together randomly with chunks of genes from the other parent. That's the process of meiosis. What would happen then if only one parent had that complex trait, and therefore only one parent had the genes needed to build that complex trait? To make a simple analogy, imagine that you had a toaster and a blender, and you broke the toaster down into parts, and you broke the blender down into parts, and you took 50% of the toaster parts and 50% of the blender parts and tried to put them together to build something. What do you think you would get, right? You certainly wouldn't get a machine that could either blend or toast, right? It would not work. With sexual reproduction, if only one parent has the genes for a complex adaptation, the offspring is very, very unlikely to inherit them all, and thus is unlikely to be able to express that adaptation themselves. And so that adaptation wouldn't actually be inherited. For sexual reproduction to reliably reproduce complex adaptations from parents to offspring, this requires the genetics of those adaptations to be reliably present in both parents. In other words, it requires that the genes for those adaptations are typical for the species, such that they can expect to encounter themselves or encounter the other genes that make up the trait in the other parent that they're meeting. So at this point, you should be thinking to yourself, well, if that's the case, how can anything ever evolve, right? And you're right. This presents a major hurdle for the evolution of complex traits for a sexually reproducing species. But the hurdle can be breached. For simple things, where a single mutation is enough to produce a reliably positive effect on fitness, that gene can spread to fixation, even in a sexually reproducing population, because every offspring that it finds itself in, on average, is going to have more kids. And so it's likelier to be passed on in greater numbers, and so that gene will spread. For anything more complicated than that, anything that requires two or more genes working together to produce the trait, for these genes to be selected by natural selection, this process has to have been repeated more or less one step at a time. Each step towards that complex adaptation that is coded for by a big set of many genes, if the gene in question only produces a positive effect, given that there's 5 or 6 or more other genes already present, for this gene to evolve, those other genes must already have been selected such that they're species typical. This is a very large constraint on how sexually reproducing species can evolve. The upside here is that we now have a powerful new tool for testing between different theories of adaptations, because adaptationist theories are going to be more plausible, more likely to be true, if you can identify each transition in the evolutionary history of that adaptation as itself an adaptation. But at this point, we also need to remember that each of those stages in that evolutionary history doesn't necessarily have to be in response to the exact same adaptive problem. Or said another way, they don't have to be adaptations for the same thing. If you wanted to give this idea a name, it doesn't really have a name, but if you wanted to give it a name, you could call it serial adaptation, as in a series of unique adaptations. My thought here is that this helps orient your mind towards these being unique events that just happen to be in a sequence. Let me give you a concrete example to help make this clear. How did bat wings evolve? How do you get from a rodent like this to a creature like this? It's really hard to see how a small change to the arm of the creature on the left would provide any solution to the problem of flight, right? Because just making a small change is not going to make you better at flying if you start out as something like that rodent. So how did it go down the evolutionary path? Our current understanding is that the first stages of selection was actually on the anatomy of their arms. And it wasn't in response to a pressure for flight, but actually a pressure for temperature regulation. Because this creature was alive on Earth at a time when temperatures were rising. And as a mammal who has warm-blooded metabolism, with fur that keeps that internal temperature inside, that poses a problem, right? The animal could overheat. One way that problem could be solved is by stretching out the skin to provide more surface area to dissipate the heat. And mutations to the genes that control bone growth are a likely candidate, right? Because bones that grew longer would make the skin stretch out more to be able to dissipate more heat. Given that greater surface area, further mutations are now operating in an environment where you are not at the state of that ancestral rodent, you're at the state of something more like that. And somewhere between selection for increasing surface area for dissipating heat, you start to get to a place where small changes make it more likely that you're going to be better able to control your flight. And that transition from the selection pressure from temperature regulation to flight gives us our series that allows the adaptations, each of those genetic changes along the way, to actually themselves be adaptations. And the point of this example is that they didn't have to all be for flight, right? Those first ones were not about flight. An interesting convergent example is actually the same that we need to explain how flight evolved in birds. The ancestor of all birds today was a group of theropod dinosaurs that looked something like this. And depending on how young you were when you went through your dinosaur stage, you maybe have already got the memo that dinosaurs had feathers, right? But they didn't use them for flight. These were not flying dinosaurs. These feathers, and they were rather primitive, they were used for display. These primitive feathers were themselves derived from scales, and they had a very simple structure in their filament that just allowed them to stand up and spread out. And that helps the creature look a little bigger, so maybe a little tougher to its rivals, or maybe a little sexier to its potential mates. But for part of the time that these creatures were around on Earth, Earth was going through a cooling phase. And now the problem is keeping heat within the body, keeping, for example, eggs that need to be kept warm, warm. A mutation to the genes that control the filament structure of these kind of early feathers could make them more dense, allowing them to trap more air, to give the creature a better ability to use her own heat or trap her heat to keep her eggs warm. Once those genes for filament structure that have been under selection for trapping heat are species typical, that presents a new genomic environment, because now new mutations that use that trapped air in an aerodynamic way, and that's all aerodynamics is, moving trapped air, means that simple changes to the shape could make them more likely to be able to help you move in a way that would enable flight. So we go from selection for display to selection for trapping heat to selection for flight. Each of the steps along the way, as an adaptation, that gives us a plausible story of how you could go from a creature that didn't have feathers and didn't fly to a creature that does have feathers and does fly. So that solves that problem. We have some clear paths as to how complex adaptations can evolve, even in sexually reproducing species. Genes for adaptations have to be universal. But I hope at least some of you are thinking to yourself, wait a second, doesn't this cause a problem for why sex evolved in the first place? If we need genes that code complex adaptation to be universal, then how can species typical genes that are in both parents lead to variable offspring? Isn't the whole point of sexual reproduction to produce offspring that are variable from their parents so that their pathogens don't have a head start in exploiting them? Wasn't that how we got to sexual reproduction in the first place? That's the confusion you should have right now. To answer what looks like a paradox here, we have to realize that the level at which we need variance to encounter pathogens and the level at which we need universality to build adaptations are different levels of analysis. To defeat pathogens, we need variance at the exact way the mechanism is built, that precise biology and chemistry where the pathogens are exploiting the host's body. For example, bacteria can enter a cell by mimicking the molecules that the cell normally uses, those proteins on the surface of the cell, to allow resources to get in. So the way that that bacteria is exploiting the cell is by mimicking those specific molecules. You can think of that as the precise physical implementation, using David Marr's terms. In contrast, for sexual reproduction to result in adaptations that can be inherited from parent to offspring, a population needs invariance at that functional level at which the parts of the adaptation works. You can think about this like the computational level of analysis that Marr identified, the level that actually solves the problem of the adaptation. Here's an analogy using a different kind of mechanism. Imagine that there's a population of car thieves and they're exploiting an auto manufacturer. That's the host. So the car thieves are the parasite and the auto manufacturer is the host. Back in the day, this doesn't work anymore. To hotwire a car, all you had to do was access the bundle of cables that contains the wires that connected the ignition switch and bypass the switch So you don't have to put in the key, because by bypassing the switch, you could trigger the car to start. Imagine that the automaker always used a red and a blue wire when they wired up the circuit. Once the car thieves figured out that all they had to do was cross the red and blue wire, they could very quickly cut into the cable, find the red and blue wire, and cross them. That way they could break into cars and steal them very quickly. But does the color of the wire affect how the wire works? Does the color of the plastic on the outside change how the wire works? No. For the circuit to work, the wire just has to connect the right parts. If the automaker randomized the colors of the wires that it used, the car thief would have no idea which wires to cross when they were trying to steal a new car. And it would take them more trial and error working through all the combinations before they got to it, maybe long enough for them to get caught by the police. The car thief was exploiting the precise implementation of the mechanism, those details that are just kind of how it works. The automaker fought back by creating variance at that level, but that variance didn't change how that adaptation, those wires, worked in the way that they're actually solving the problem that allows the car to run, right? That allows electricity to go through the ignition switch. That is, it's invariant at the computational level of analysis. We can see an example of this exact phenomenon in humans. And one of the kind of simplest cases is with blood types. You probably know that there are different types of blood. In fact, this is a very simple scheme. There's actually like 87 different schemes for classifying differences in blood. But the most common that people know is the ABO system. And you might even know your own blood type. Some people are type A, some people are type B, some people are type O, some people are type AB. So we have different kinds of blood. And for you, your blood works for you, for me, my blood works for me. All of our blood works fine in our own circulatory systems. But that variance seems like variance to pathogens. In fact, it's actually the pressure of pathogens that causes that variation in blood type to be maintained over time, because pathogens impose frequency-dependent selection on blood type. In contrast to a case where selection is always going in the same direction, selection is frequency-dependent whenever the direction flips based on the frequency of the trait. In particular, selection is frequency dependent when, if the allele is under positive selection when rare, meaning that when it's rare, it's going to increase in frequency to become less rare or more common, but then becomes under negative selection when it's common, meaning that it gets less common or more rare. That's going to lead to mixed or cyclic equilibria. Or you can think of this as a rock, paper, scissors dynamic. If I always threw paper every time, you could quickly learn how to beat me. But if you don't know what I'm going to throw, because sometimes I throw paper, sometimes I throw scissors, sometimes I throw rock, you wouldn't do as well. And so I need to maintain each of these strategies over time, because if I throw paper a whole lot and that starts to not be good anymore, the first time I throw scissors, it's going to be a really good strategy, and it's going to be better for me to do that. We actually already simulated a case of frequency-dependent selection when we explored the hawk-dove game. To refresh your memory, the hawk-dove game was when we had two organisms that met over a resource that they both wanted, and they have to choose whether to fight or not without knowing what the other would do. And fighting is costly, so if you lose, you get hurt. The success of strategies in this game is frequency-dependent, at least under some parameter combinations, when the following is true. If there are more hawks in the population, then it's better to be a dove, because at least that way you don't end up hurt. Then this is going to cause the frequency of doves to increase when doves are rare. But when doves get common enough, now they're most likely to encounter themselves, and the most likely thing to encounter is a dove. And in that case, the direction of selection switches. Now it's better to be a hawk, because you get the sandwich, you get the whole sandwich instead of just a half a sandwich. So doves increase when rare, and hawks increase when rare. That's what led to a stable equilibrium where you had both hawks and doves in the population in a mixed rate. It wasn't that hawks went to fixation or doves went to fixation. It stayed in a mixed state because selection was frequency dependent. Okay. Finally, let's look at another real example with a specific pathogen, the parasite that causes malaria. This pathogen exploits one of the molecules that make up our blood called hemoglobin S. This is one of the components that we use to build our blood cells. The malaria parasite has specialized in exploiting the common version of this gene. So if you have the common version, you're more likely to end up getting infected with malaria if you have an exposure. Now, since humans are diploid, meaning that we have two sets of chromosomes, one from each parent, you actually have two spots, one on each of those paired chromosomes where you carry these alleles. If you inherit the common allele from both parents, that is, you're called homozygous for it, you're particularly susceptible to malaria. Homozygous means the same for those alleles. But mutations that change the biochemistry of hemoglobin S make you more resistant to malaria. And so when those mutations are rare, they're under positive selection. And if you only have one of them, you have what's known as sickle cell trait. You have the ability to have a child that has sickle cell, but you yourself don't have sickle cell. You just carry one of the genes for it. It makes you malaria resistant to have one copy of the common type and one copy of the rare type. The problem is that once that rare type gets common enough, it's likely to meet itself and become homozygous, meaning you have two copies now of that mutation. And that's what ends up with sickle cell anemia. This is the case where what normally your red blood cells are round. If you have sickle cell anemia, they make these sickle shapes that cause them to be much more likely to form clots and cause problems in your circulatory system. So what's the selection pattern for the mutation? When it's rare, it's hardly ever going to meet itself. It'll almost always result in an offspring that's heterozygous and therefore doing well, so it'll be under positive selection. But once it becomes common enough, it will be more likely to meet itself, right? It'll be more likely to experience sickle cell anemia and be under negative selection. This example of frequency-dependent selection is an important case to remember, because without thinking about this frequency-dependent effect imposed by pathogens, it would be hard to understand why sickle cell anemia hasn't been simply removed by natural selection, right? Why does it keep sticking around? It would be hard to interpret why some places in the world have a higher rate of sickle cell anemia than others. Turns out it's those places where malaria is common. This gives us the kind of textbook example of that push-pull relationship between natural selection on the functional organization of a species and that selection pressure induced by pathogens to maintain diversity at that biochemical level. Sometimes that later selection pressure, the one to maintain that kind of diversity, it can be so strong that it even balances out where some proportion of the species ends up with the disease, because the odds of having that disease kind of balance out against how good it is to simply be heterozygous for it instead of homozygous for it. The advantage you get from having the variance might counteract the disadvantage you get from having the disease. And that's going to maintain that disease state in the population over time, even if it's itself under negative selection. So pulling together what we've learned so far, we can describe our current understanding of human nature like this. We expect human nature to be made up of adaptations that are universal in our species, at least when you characterize them at the computational level of analysis. And on top of that universal architecture, we expect there to be variance in human nature that's caused by frequency-dependent selection that maintains the variance over time because of pathogens and other sources of frequency-dependent selection. But is that the only source of variance that we expect? Is it really the case that we expect universality for absolutely everything else? Let's take a few steps back and remember how we got here. Part of our causal model of why adaptation should be universal is that natural selection is eliminating all of the functional variance in those adaptations. Anything that was functionally contributing to the adaptation working more poorly than an alternative, that version that works more poorly should be under negative selection. But not all of the traits of an organism are going to affect reproduction, right? Not all of them are going to be adaptations. Going back to our allegory of the blind men and the elephant, right? There's an infinite number of ways to carve up any species into different kinds of traits. And most of those traits are not going to be the traits that were actually the target of natural selection. We're going to need some new concepts, some new terms here. We can define an adaptation if it was the solution to an adaptive problem. That is just the same lock and key definition that we had before, right? That trait's design is best understood at the computational level of analysis. But of course, it's going to have an implementation, and those precise details of the implementation give us another category of traits that we can call byproducts. Byproducts are traits that are themselves not the computational solution to an adaptive problem, but they're simply the result of the implementation to such a solution. This is like, because we need to have mechanisms at the boundary of our cell that bring resources in and out. As a byproduct of that, we have these proteins that allow bacteria to exploit us. That's going to leave a small residual of traits that don't meet these criteria, which we're going to call noise. Noise are those traits that are not reliably associated with the computational solution to an adaptive problem. They just vary kind of independently. So those are the abstract definitions of those terms. Here's some more concrete. Looking back at our circulatory system, we can find traits that are clearly adaptations, right? Our blood has that molecule hemoglobin that's able to bind oxygen and move it through our body to where it's needed. The fact that hemoglobin uses iron to do that, and that when iron is oxidized, it looks red to our eyes, such that when our blood is oxygenated, it looks red, that trait is a byproduct. Why? Well, there's no evidence that this trait solved an adaptive problem, right? There's no evidence that having blood that was red as opposed to some other color meant that our ancestors had more kids. It can be explained merely as the implementation of how the adaptation was solved, how oxygen got bound and moved using this molecule that contains iron. Notice that these two categories both contain reliable features of our species. Our species has both of these traits. The category that doesn't meet this criterion is noise. If you looked in my body, you would find at this moment a particular number of blood cells that you could count, and it would differ from the particular number that you could count in somebody else. That variance from person to person is the hallmark of the noise category. Noise just varies randomly. Considering these three categories of a trait, adaptations, byproducts, and noise, we can go through them and see how they can lead to predictable variance in human nature, and also predict which pieces should be universal. So adaptations are those reliably occurring traits that have their computational design because past selection selected that trait, shaped that trait. This is going to be the source of most of the universality within a species. Byproducts can be just as reliable and universal as adaptations, but we can't explain their design as part of the computational solution to an adaptive problem. So selection, in a sense, is responsible for the trait because it's part of the implementation to an adaptation, but not because of the design features it has per se. And we can think of different subcategories of byproduct like that. So one type is simply the implementational details that are being dragged along with the trait under selection, right? This is like how our blood turning red when it's oxygenated gets dragged along with the adaptation of carrying oxygen. The trait redness isn't being maintained by selection because it's useful. Rather, it's simply a byproduct of how hemoglobin works and how hemoglobin is useful. Other traits are obvious byproducts because we can use adaptations that got selected for one thing outside the scope of the conditions that selected for them. For example, as animals that walk on two legs, we have adaptations for keeping our balance on 2 feet as we stand or walk or run. And because we have those adaptations and the fact that we live in a world that now has skateboards, some of us, not me, some of us can use those adaptations in order to skateboard. That is, these adaptations also have the trait allow to skateboard. But this trait is clearly a byproduct because skateboards were not part of that ancestral past where that selection was happening. None of us have traits. None of us have the genes we do because our ancestors skateboarded better or worse, right? Therefore, that trait, ability to skateboard, can't be an adaptation. And third, some of these traits are simply reliable ways that adaptations break down or malfunction. For example, we have adaptations for choosing what food to eat. And these mechanisms evolved in a world where sugar calories were very scarce. Finding things that contained a lot of sugar was a rare event, and when you found it, you had to eat it. In such a world, traits that made it very likely that you would eat all the sugar you could if it's available would have been a good design feature for the mechanism. But in the modern world, we live in a very different situation where sugars are plentiful. And now our strong preference for sugar reliably leads to acquired or type 2 diabetes in our modern environment. This ability to have type 2 diabetes, or the commonness of type 2 diabetes, is a reliable trait of human nature, but we wouldn't say that it's an adaptation. Rather, it's a byproduct of one way that our diet choice mechanisms malfunction, or because they're mismatched to the modern environment. So this is another way that byproducts can result in individual differences. In some places, you'll see diabetes more than in other places. That's variance across humans. And so because these byproducts are part of that implementation, those implementation details are going to interact with different features of the environment to create differences in the trait, causing individual differences. And finally, we have noise. These are those traits that are not reliably associated with adaptations. Like, you have a precise number of hairs on your head and it's different from the person next to you, or you have a precise fingerprint pattern that's different from the person next to you. Genetically, noise is the result of novel mutations, new mutations that natural selection hasn't yet touched, or mutations that don't have any effect on reproduction, and so therefore natural selection doesn't touch them. This is actually a pretty big source of individual differences, and let's see why. If we think about these mutations in the context of selection pressures, we're going to see that this creates kind of like a residue of individual difference. Imagine a gene, and this is the formula for the effect I'm going to show you. Imagine a gene that's under positive selection because how it contributes to an adaptation. Every generation, we experience a small rate of mutation such that each of our genes has a chance of mutating. Modeling a simple example of that, we can imagine that this gene is under a small force of selection, say 1.5%. That's that 0.015 in the term. And it also experiences some rate of random mutation that sometimes pushes it up and sometimes pushes it down. Running this model over time, we can see selection pushing that trait up to fixation, but mutation perturbing the fixation, pushing it down. And that means if we look at the average value in a population over time, we can reliably see that there's a small residue of individual difference. Some people have the mutation always over time, because that's the balance point between selection and mutation pushing in opposite directions. That point where mutation and selection balance out depends on the relative strength of mutation rate and selection. Compared to this model, where selection was a 0.015, in this model, selection is a little weaker. It's only a 0.01. And when selection is weaker, we can see that the average point of mutation and selection balance is leaving a bigger reservoir of variance. And that's all due to the weaker force of selection that's pushing against mutation. Finally, we can think of this in a bidirectional way. This is often called balancing selection, where the fitness maximizing level of a trait is at some intermediate value. You can think of this like height, like there's positive selection as you're getting taller and taller and taller, up until the point where if you were taller than that, it would cause problems. And then it's worse for you to get taller and taller and taller and taller than that. That's what maintains height at kind of a intermediary value where, like, we could have been taller, we could have been shorter, but we're maintained at kind of that intermediate height. We can model that with an equation like this, which flips the direction of order depending on the relative position. But even that kind of model, it's important to remember that that has mutation in it, right? And so it's not that our population maintains at an exact perfect 50%. This is flipping around on each side of 50%. What's happening here is that selection is pushing on both sides towards the middle, and mutation is pushing back on both sides. right? And so it's creating a residue of individual differences within that range. That's why we have variance in height that's typically within this kind of middle range, as opposed to equal amounts of variance as you get shorter and shorter and shorter and shorter, and taller and taller and taller and taller. Returning to our view of human nature that we checked in on at the top, our new model says that we expect adaptations to be universal and species typical, except when they're frequency dependent. And we expect more individual differences because they're the result of byproducts, and also that reservoir of variability that we get from mutation selection balance due to noise. Still, though, that doesn't really seem to cover a bunch of big individual differences, right? People differ in all kinds of ways: intelligence and personality and strength and verbal ability and kindness and anger, on and on and on. And it doesn't seem like all of these are just byproducts or frequency-dependent selection and noise, right? A lot of these things seem like what natural selection would act on. Probably not byproducts. Probably not frequency dependence or noise-driven mutation load. Is there some other category of individual difference that we're missing? To prime your intuitions of what that something might be, consider the following thought experiment. Imagine that we have a smartphone that has a GPS mechanism and a folder of MP3 song files. And it has an app that chooses the song to play based on the readings of the GPS, the latitude and longitude. If we put identical copies of that smartphone around the world, latitude and longitude vary around the world, we would see that there would be individual differences, right? Smartphones in one place would be playing one song. Some of those smartphones would be playing a different song if they were in a different place, and so on. But we also see similarity within a region, right? Smartphones in the same place tend to be playing the same song. But not because they were talking to each other, not because they have any interaction with each other at all. But this is exactly the pattern that we call culture, right? That individuals in the same place have the same habits, preferences, norms, behaviors, speak the same language, etc. We often think that that's because of interactions between people, but from this example, we can see that it doesn't have to be that way. You can have people ending up the same, not because they coordinated or communicated with each other. All right, that is where I'm going to stop for today. I think I have time for one or two questions. Any questions going back to heritability or serial adaptation or all of these mechanisms of individual difference? Daryl?
Oh, yes. Since it talked about pathogens, I'm thinking about the coronavirus. Right now, we have so many variants of the original coronavirus. Is there a heredity, like heritability, high heritability between different variants of the original coronavirus?
So I don't know of any studies that look at if susceptibility to a particular variant runs in families, but you would probably imagine that for a brand new virus that our species hadn't been really exposed to before, that virus wouldn't necessarily have, like that variant wouldn't have exploited a particular thing that runs in families, such that natural selection would then act on varying up that thing. But that is what's explaining the evolution of the variants. As the variants have evolved, they've gotten better and better and better at fooling our immune system.
Right. And they become more contagious. We have been vaccinated, but these new variants are able to, somehow counterbalance the effects of the vaccines.
Yeah, so the way that, so our immune system uses certain features of the outside surface of a cell or a molecule to recognize a like dangerous pathogen that they need to encounter. And what the variants have changed is exactly those molecules on the outside, what they've called the spike proteins, those surface features that our immune system like uses to detect that it's this virus. And so it's basically like putting on a disguise. It's saying like the immune system is looking for the thing that it saw before. And now the new version is basically like wearing makeup and pretending to be somebody else.
Right. Thank you so much for answering my question.
Yeah. All right. We are over time. Hope you enjoyed today, and I'll see you next week.